Good morning, everyone, and welcome to today's eChalk Talk, sponsored by Sapient. We are very lucky today to have Dr. Mohit Jain. Dr. Jain is a physician scientist with nearly 20 years of expertise in physiology, biomedicine, engineering, computational biology, and mass spectrometry-based metabolics. Prior to founding Sapient, he formed and was director of Jain Laboratory at the University of California, San Diego. There, he led a multidisciplinary research team of chemists, engineers, mathematicians, epidemiologists, and physicians to develop next-generation rapid liquid chromatography mass spectrometry systems to probe the non-genetic landscape of disease across population-scale human studies. His work was supported by the NIH ONES program grants and over $30 million in federal, foundation, and industry funding. Dr. Jain founded Sapient in 2021 as a spinoff of Jain Laboratory to expand upon the mission of accelerating human discovery and drug development through the nexus of high-throughput analytical mass spectrometry, computational biology, and population-scale clinical studies. It is my pleasure to introduce Dr. Mohit Jain. Dr. Jain?
Thank you so much for the warm introduction, and I appreciate everyone's time today. We're excited to be representing Sapient here and hope to spend the next hour or so, or 45 minutes, I should say, sharing with you some of our experiences in the use of large-scale mass spectrometry to accelerate biomarker discovery and drive drug development. And so I'll go ahead and jump in here, and I look forward to your questions as we go through this process. Okay, so the main challenge here that we're faced with at Sapient is very much the same one that many of you are combating every day in biopharma, and that's the abysmal failure rate that's associated with drug development. Now, there's a number of statistics that you can sort of quote around this, but most folks agree that somewhere upwards of 90% of all drugs that enter into the pipeline ultimately end up failing. And as you can imagine, there's a number of consequences that emerge from this. Now, we spend quite a bit of time thinking through why drugs fail and why less than 10% of these assets actually go through to the clinic. And one of the key reasons is what we think of as this fundamental mismatch between how we defined disease, which is really based upon an end pathology, and how drugs are developed, which is really to target an underlying mechanism of disease. And so the mismatch between defining disease and developing drug targets is really one of the main issues that compounds this failure. Now, as mentioned, there's obviously a number of consequences that emerge from having this type of failure, both in risks to patient, and obviously there's a very high risk of severe adverse events, time delays, and 10 years being a conservative estimate for how much these delays can consist of, and ultimately expense to your organizations. And there's various statistics around here, but this is upwards of the hundred millions to billions of dollars in lost revenue. Now, as mentioned, the real diagnosis of disease has not changed in the last several thousand years. And shown here is the urine wheel on the left and a pathologic specimen on the right. And fundamentally, this is still the way in which we diagnose disease today, which means, again, based upon end pathology with very little understanding of the underlying mechanisms or substratification that gives rise to disease process, which is, again, how we begin to target drugs. Now, here at Sapient, we really very much believe in biomarker-driven drug discovery, and we view biomarkers as the ability to overcome this underlying mismatch. Now, fundamentally, what biomarkers allow us to do is to peer into the individual and begin to align specific patients with their specific disease state, meaning the mechanism of disease for them, with a specific drug therapy. And this can take many, many different sort of angles and flavors, but with regards to patients, we can stratify individuals who are most likely to respond. With regards to disease states, we can improve early detection and predict progression. And ultimately, with drug therapies, we can optimize dosing and timing, and again, beginning to identify the right individual at the right disease point for the right drug. Now, there's a tremendous amount of data and evidence in the clinical literature over the last 2 to 3 decades that biomarkers can absolutely transform drug development. So this is an analysis that was performed independently of us across over 20,000 clinical trials across a number of different therapeutic areas. And there's one universal lesson that continues to emerge from these types of analyses. And that is those assets, whether they be small molecule, large molecule, gene and cell therapy, irrespective of the modality, those assets that are developed together with a biomarker have somewhere in the order of 2 to 10-fold increased FDA approval. And now again, this is across a variety of different therapeutic areas, from oncology to cardiometabolic disease, autoimmunity, immunology, vaccine development, et cetera, et cetera. And not only is the FDA approval rating higher relative to those assets that are developed with a biomarker, but the time to approval is much faster, on the order of 20% or greater. And as you can imagine, these types of numbers can absolutely transform a drug development pipeline as well as a drug development organization. Now, again, there's nothing magical about biomarkers, but essentially they allow us to identify and confirm the right underlying biological mechanisms, the right underlying target, including target engagement, the ideal safety profiling, the right pharmacometrics, again, dosing, timing, and interactions, and ultimately the right patient, aligning the individual with their specific disease, with this specific target, and ultimately the specific drug or disease intervention. Now, when we think of biomarkers, oftentimes our mind wanders to genetics, and certainly the area of genetics and genomics has transformed biomarker discovery and drug development in the oncology space. But genetics alone is not sufficient to decode human disease. Now, if we do a broad scale analysis across most adult onset diseases, once again, whether it be cancer, heart disease, neurodegenerative disease, lung disease, metabolic disease, autoimmune disease, et cetera, what we find is that at broad scale, somewhere in the order of about 20 to 30% of population attributable risk is due to genetics, which means that over 70 to 80% of population attributable risk is not captured by the human genome. And we know this. Genetics is set from the moment of conception, which means everything that occurs after the time of birth, everything about the way you live your life, everything you eat, drink, smell, smoke, what you eat, etc., that information is not encoded in your underlying genome. And that information could have tremendous influence on health and disease and drug responsiveness. Now, it turns out that these other factors, whether they be dietary, drugs, internal organ communication, meaning how your liver communicates to your brain, how your fat cell communicates to your heart, environmental exposures, the microbiome, both skin and gut, as well as toxicants, and many, many other factors about the host, the underlying disease, and the world in which they live, again, is not encoded in underlying genetic sequence, but is actually encoded in small molecule chemistry. Now, these are the thousands of small molecules that travel around in our blood, originate from various sources, whether it be diet, the microbiome, our liver, our heart, etc., and signal to distal organ sites where they influence human disease over time. Now, traditionally, these small molecules or this level of information has been captured by a technology called mass spectrometry. And the real challenge is that while mass spectrometry is quite robust, and there's been a number of stepwise advances in mass spectrometry over the last decade or so, it's still too slow for population level discovery. Now, this is where Sapient emerges, and we very much view ourselves as a discovery engine. There are 3 pillars under which we operate to advance biomarker discovery and drug development. The first is an underlying analytical technology that's shown here on left. This is a next generation proprietary rapid liquid chromatography, or RLC, mass spectrometry system that enables population level discovery. Essentially, what these technologies allow us to do is to take a small amount of sample, whether it be blood, urine, CSF, tumor, tissue, cells in a dish, all the way down to soils and microbes. And in that very small biosample, we can assay somewhere in the order of 11,000 to 15,000 biomarkers. And we do this quite rapidly, in about 42 to 43 seconds analytical cycle time per sample. Now, we capture broad chemistries, a number of small molecule factors below a molecular weight of 2,000, not 200, but 2,000, including biologically relevant proteins and thousands of yet unmapped factors. Now, this entire pipeline is fully automated through freezers and sample excisioning, handling, processing, and mass spectrometers are completely robotically controlled. And so Sapient is one of the largest, if not the largest, capacity bioanalytical discovery facility in the world, routinely analyzing over 20,000 biosamples per day. Now, the second pillar that's shown in the middle here is, as you can imagine, as we're generating these thousands of terabytes of data at a time, analysis and understanding of these types of data is very complex. And so as part of Sapient, we have a very deep biocomputational learning team, which has deep expertise in the handling of mass spectral data, as well as the integration of mass spectrometry biomarker data with preclinical information, other biomarkers, genomics information, the microbiome, human biology, circulating factors, inflammatory cytokines, and ultimately clinical outcomes. And so this team is able to apply tools as regression statistics all the way up through machine learning and AI-based approaches to begin to identify key molecules and key biomarkers and derive very actionable insight from very complex multidimensional data. Now, the third asset or the third component of Sapient is what we call an underlying biological database. And essentially, this is a proprietary human data repository that Sapient has collected that now includes over 300,000 people from around the world in which biosamples have been obtained, and these samples analyzed using our analytical technologies. And in each of these individuals, we have longitudinal outcome over 20 years of time, including adjudication of clinical outcomes, demographic features, lifestyle factors, dietary information, drug response, human genetics, microbiome, cytokine levels, et cetera, et cetera. And so this is an incredible rich repository from which we can begin to make actionable insights through mining. Now, there's many ways in which we can operate across these three pillars, starting with biological samples, making these analyses, doing computational learning around them to identify key molecules, and then cross-validating of observations in our biological database, as well as we can go in the reverse direction by which we use our database and mine this information to derive new biomarkers that subsequently we validate using biological samples. Now, as mentioned, the real goal here is discovery at scale, not in dozens or hundreds of samples, but hundreds of thousands of samples at a given time. And this is really the nexus at which Sapient begins to operate. Now, to give you some idea of what we can actually learn from these approaches, shown here is an analysis across 50,000 diverse individuals from around the world. And what you find is on the left here, while we can capture 11 to 15,000 molecules per person, not every individual has exactly the same molecules in their blood as would be expected. We all eat slightly different things, we live in slightly different areas, we're exposed to slightly different things, etc. And so across these 50,000 individuals, we capture somewhere in the order of about 180,000 circulating factors across the entire group. Now, when we look across these 180,000 circulating factors, as shown on the Venn diagram on the left, we find that somewhere in the order of 10 to 12,000 of these molecules in blood are what we'd consider common, meaning they're present in greater than 50% of the population, irrespective of where the world you live, your age, your gender, your other lifestyle factors. Among those 12,000 factors, there's somewhere in the order of about 3,000 of these factors that are universal, meaning every individual has these molecules in their blood. And this includes the common sugars, nucleotides, lipids, proteins, amino acids, et cetera, that are required to walk and talk and live in the universe. Now, if we look among these 12,000 molecules, or I said, if I take a step back, if we look across these human biological samples, we often find that humans have very much defended the number of molecules that are in human blood, and this has to do with maintenance of osmolarity. And again, you can see this distribution here that's shown on the top left. On average, an individual has somewhere in the order of about 11,000 to 15,000 molecules in blood. We can find certain extremes of populations, individuals that are extremely young, meaning newborns, or individuals who are extremely old, centenarians, who tend to have slightly more molecules in blood. If we look at the top right figure, we find that there clearly is a distribution once again in what's shared among individuals in an absence presence. And this goes back once again to the fact that not all of us have exactly the same molecules in our body. If we could look at the bottom left, we can begin to understand not only the absence presence, but the dynamic range of these molecules in our blood. And when we look across the 12,000 common molecules in individuals, we find this very interesting pattern by which there's somewhere on the order of about 1,000 or so molecules that are highly stable in individuals. This includes molecules such as glucose, and whereby we all ate several different things several hours ago, assuming none of us are diabetic, our glucose levels are all maintained and defended within about 5% of one another. And that would be such an example of a very stable, well-defended homeostatic marker in blood. On the other hand, we can look to the right of this peak and we can see there's somewhere in the order of several thousand molecules that are hypervariable, whereby while we all have these molecules in our blood, their concentrations can vary by orders of magnitude from individual to individual. These include molecules that are less homeostatic, that have incredible predictive power for understanding human health and disease over time. We can also begin to look and see how molecules change over time. Certainly molecules in blood are quite dynamic, and if we look at molecules in individuals every hour on the hour over a 24-hour period, we find that more than 50% of molecules change more than 50%. Now, this, of course, has to do with very key factors such as sleep-wake cycles, fasting and feeding cycles, whether or not you're running around after your kids or sitting down on the couch. And as you can imagine, human chemistry and human markers change substantially over that time frame. However, if we all sample our blood today under a fasted first and morning condition and come back several years later, shown here is over a 2-year time period, and sample our blood again first and morning under a fasted condition, we find that molecules change actually very little, and less than 50% of molecules change more than 20%. And this continuity has to do with continuity exposures. We tend to eat the same things, we tend to live in the same places over a several-year time period, and our bodies tend to function in the same way. And this consistency and stability in our fasting chemistry is what allows us to use these markers for drug discovery and biomarker discovery. Now, we can begin to ask, what are the factors in humans that influence these markers in human blood? And in doing so, we can begin to take several hundred to several thousand mono and dizygotic twins, and you can statistically calculate the amount of influence that comes from genetics versus environmental factors versus shared environmental factors. And when you do so, you find is that in the environment in which we live is an absolute key determinant of the molecules that are in human blood. And again, this is a way in which we can begin to map what we would consider to be the non-genetic landscape of human disease. These environmental factors include such factors as the microbiome, as shown in the middle panel here, that has a huge influence on molecules that are in circulation. And we find that over 1/3 the molecules floating around in humans today actually are microbial in origin, and a number of these we've mapped back to specific bugs within the gut microbiome. We also find that other dietary, lifestyle exposures and laboratory values can have profound influence on circulating molecules, as shown on the right here. And I recognize these are written in small text, but essentially everything from age, gender, BMI, race, socioeconomic status, geographic location, blood pressure, degree of exercise, et cetera, all of these factors can influence circulating biomarkers. And by making these singular measurements, you can begin to provide insight into each of these risk factors and how they influence disease over time. Now, when we think about the circulating markers that are in blood, chemically, they're incredibly diverse. And shown here are just some of the common molecules that we can capture. Typically, from a scan of several thousand individuals, once again, we find that on the order of 11 to 15,000 molecules are present in any biological sample. Among those, we can readily identify somewhere in the order of about 1,000 to 1,200 of these molecules, which means the vast majority of molecules floating around in humans today have actually never been mapped. Now, there are reductionist chemistry approaches that we have in the lab by which we can isolate these unknown molecules, concentrate them, and solve their structure using NMR and other cryo-EM-based tools, and ultimately synthesize them to identify the chemical structure of unknown molecules. Now, I think technology is wonderful and data is always exciting, but really the goal here is actionable insight. And so I'm hoping I can take a couple minutes to share with you some real-world applications by which we leverage these technologies and we leverage Sapient's computational tools and back-end biological databases to begin to make some fundamental observations and discoveries that advance forward medicine. And so what I've done here is just selected several cases across a variety of different disease areas that I'm hoping we can spend a couple moments just to review together and think through how these approaches may be helpful. The first here is around a human disease known as NASH, or non-alcoholic steatohepatitis. For anyone that's worked in the human metabolic disease space, you may be well aware that NASH is an incredibly prevalent disease, similar to diabetes and obesity, but is incredibly complex to diagnose. And that diagnosis of NASH typically requires invasive liver biopsy with a large bore needle, which is more grotesque than even it sounds. And in doing so, it's been very difficult to enroll patients in clinical studies with NASH, as well as follows these individuals that typically require, again, serial biopsy. And because of this, there's been a number of challenges with clinical studies and drug development in this space. And so what we did here at Sapient is we began with a cohort of approximately 10,000 individuals, which is shown here on the left. We assayed their blood using our high throughput mass spectrometry systems, measuring over 50,000 molecules in these individuals that are shown by the little gray dots on this volcano plot here. And we simply asked across these individuals, are there particular molecules in human blood that are reflective of the development of underlying liver disease, in particular NASH? And what you find is that there's a number of molecules that are highly increased in individuals that go on to develop liver disease, which are shown by these little red dots. What I can say is all of these molecules are unknown chemistries or unknown molecules that have never been mapped anywhere in the world. And as we begin to look at these molecules, even in the absence of structure and deformation, we can tell you that 3 of these molecules that are shown here by these blue circles are chemically very related, though independent of one another, and likely lie in the same biological or chemical pathway, which is shown in the middle figure here by this networking analysis plot. Now, among these molecules, we can tell you once again, they lie in the same biological pathway, and we can prioritize one of these molecules that's labeled here appropriately as key biomarker. And we can begin to validate this key singular biomarker now in preclinical as well as human studies. Shown on the upper right here is a preclinical study. This is a mouse model that develops NASH versus the precursor disease state, which is termed NAFLD. And what we find is that mice that develop NASH have a very marked elevation in this molecule that starts very early on in the disease process, even before there's pathologic evidence on biopsy of liver inflammation. Interestingly, those mice that develop the precursor disease state, which is known as NAFLD, which is a relatively benign clinical state, actually have 0 level of this molecule in blood. So this really represents an all or none troponin-like biomarker that can be used for isolating out NASH versus NAFLD patients. Now, when we look across biopsy-proven human disease studies, which is shown on the bottom right, what we find is that with increasing stage of severity of NASH, the molecule in blood continues to increase from stage 0 to 1 to 2 to 3. We also see this very interesting phenomenon that's consistent across many complex human diseases, in even the most advanced NASH individuals shown on the right, whereby there are likely subsets of individuals. One subset that has relatively low levels of the molecule, and another subset of individuals that have a very high level of the molecule. Now, these individuals probably represent different biological pathways by which they develop NASH. And this is extremely important in thinking about stratification of patients in your clinical studies and enrollment of specific individuals who are most likely to benefit from a particular therapeutic intervention. We can also begin to ask how this biomarker operates over time and how early on in the disease process these markers can be increased. And when we look across Sapient's human biological database, again across over 100,000 individuals, we find that individuals that have a higher level of the molecule relative to those individuals that have a low level of a molecule are 5 times more likely to develop clinically apparent liver disease over a decade-long period. And so this is a biomarker that's elevated not only in a subset of individuals with severe disease in a case control manner, but also predicts individuals up to a decade in advance who are likely to go on to develop liver disease. Now, you can imagine from a drug development perspective, being able to intervene at a very early disease perspective and point can be absolutely critical for a number of different diseases, including for oncology, neurodegenerative diseases, and for liver disease, as shown here. We also find that this biomarker is highly dynamic, and interventions that improve hepatic fatty infiltration and inflammation result in a decrease in the actual biomarker. So it can be used as an intermediary marker to read out actual hepatic processes. And shown here is the intervention of bariatric surgery, which results in very rapid improvement in liver fat and liver inflammation. And what you can see in these individuals in these human studies, that the marker acutely declines. We can cross-validate this once again in the preclinical models of NASH and NAFLD, by which when the mice are taken off a high-fat diet and their liver fat improves, the marker once again acutely declines, making it an ideal dynamic marker for screening of early assets in NASH development. Now, we can also do advanced analyses around this singular biomarker to begin to understand where it comes from and how this may actually give rise to different disease processes. So what's shown on the left here is data that emerged from our biological database, once again, in which the molecule was assayed in over 10,000 individuals, in which human genetics information was available to us. And when we do the genome-wide association study for this singular biomarker, we can identify the key biosynthetic pathway and transport system that gives rise to the molecule, as well as understand the actual tissue and organelle from which the molecule emerges from. Now, in the case of this singular molecule, this becomes absolutely critical in that we can reveal a potential drug target that's now under development for NASH. Now, oftentimes when we do these types of analyses, causation is obviously not inferred from simple correlation, and it's important to understand whether a marker is just a passive readout of a disease state, which is in itself extremely important, or is actually giving rise to the disease itself and represents a therapeutic target in addition to being an excellent marker of the disease. Now, one way we can begin to do this is through a statistical test termed Mendelian randomization, which is a mechanism by which we can use statistics and using genetic information to infer causality. And when we do that around this singular marker, essentially looking at genetic variants that associate with the marker and asking how do those genetic variants associate with liver disease across 500,000 people, what we find is that there's this linear relationship between markers that increase, or genetic variants that increase the level of the marker, as well as increased risk of liver disease. And when you see this type of relationship, this is highly indicative of an actual causal relationship between the actual marker and the disease process. So in this case, this NASH biomarker that's been discovered has not only been cross-validated in preclinical as well as biopsy-proven disease states, not only found to be a dynamic process that can be used for screening, as well as an early disease marker, but also likely represents a new biological target whose pathway that can be intervened upon as a NASH therapeutic. Now, this is just one such example. I'm hoping I can share another example with you from the world of immuno-oncology and immunotherapy. And one of the great challenges that we face in this space is while these drugs have transformed the treatment of oncology or the oncologic space, what we find is that over 1/3 of individuals that receive these checkpoint blockade inhibitors actually go on to develop what's termed a severe autoimmune reaction. And these severe autoimmune reactions can actually limit dosing of the drug and often requires individuals to come off the underlying therapy, which can have profound influence on the overall outcomes associated with these drugs. And so we've been particularly interested in trying to identify a circulating marker that identifies individuals who are at risk for these severe autoimmune events in the setting of immunotherapy. Now, the way we started this underlying study with a client of ours was to take several clinical trials here totaling over 300 individuals who received a variety of checkpoint inhibitor drugs. And we measured over 50,000 molecules in each of these individuals, or across the entire group of 300 individuals, as shown here on the left in this volcano plot. And what you can see is there's a single marker that's shown here in red, that's highlighted, that we now have prioritized from these early analyses. And this is a marker that indicates individuals that are likely to develop a severe autoimmune reaction in the setting of immunotherapy. When we take these markers and validate them in independent studies, what we find is what's shown here on the top right, whereby in individuals that receive, for a variety of solid tumors that receive checkpoint inhibitors, these individuals that do not develop an autoimmune reaction have no change in the molecule over time. Individuals that develop a mild autoimmune reaction, whether it be mild colitis or mild rash, have no change in the molecule over time. And individuals, in contrast, that develop a severe autoimmune reaction, particularly one severe enough that necessitate coming off the actual drug, we find that these individuals have a precipitous decline in the actual biomarker that precedes the development of the autoimmunity. Now, we can also cross-validate these observations in a preclinical model, as shown on the bottom right. This is a humanized CTLA-4 mouse that, when given a CTLA-4 checkpoint inhibitor, actually goes on to develop severe autoimmunity and colitis. And what we find in this preclinical model is those mice that have lower levels of this circulating marker have much higher autoimmunity and toxicity, as shown by this relationship, confirming and validating the observation that was initially made in humans. We can once again begin to ask in our biological database, how does this marker associate not only with cancer-induced immunotherapy-induced autoimmunity, but also garden variety autoimmunity, including lupus and RA. And what we find is individuals that have low level of the marker relative to individuals that have high levels of this biomarker are much more likely, almost twofold higher risk of developing an autoimmune disease over a 10-year period. So once again, this is a marker that's altered quite early in the underlying disease process. And there's a ton of biology that we've done on this underlying biomarker to understand its actual disease pathogenesis and how this gives rise to this underlying association. And the long and the short of it is we found that this marker is causally related to the development of neutrophils. So individuals that have lower levels of this marker have higher activated circulating neutrophil counts, and this marker is actually involved in neutrophil development. So whereby this is another example by which we can start with a human clinical trial, identify a key marker here of safety, cross-validate that in independent studies, both in humans as well as a preclinical model, understand the relation of a drug-induced autoimmune reaction here relative to autoimmunity or non-drug related autoimmunity, and then understand the underlying mechanisms that give rise to this relationship. And this is another marker now that's being developed both as a biomarker as well as a therapeutic for individuals with autoimmune disease. Now, the final example I was hoping to share with you today is around a biological process. And so we spend a lot of time here at Sapient also trying to identify very interesting markers that can potentially reveal individuals and certain biological processes in humans. We've been particularly interested in identifying a biomarker that reads out systemic mitochondrial dysfunction. Now, the mitochondria is a fascinating organelle that's been implicated in a number of human disease states. And one of the real challenges in this area of drug development is that it's been extremely difficult to assess mitochondrial activity and mitochondrial total biosynthetic capabilities in vivo in humans. And typically this is done through indirect means such as measuring mtDNA levels, which given heteroplasmy can be highly variable and are very poor proxies for total systemic mitochondrial function. Or typically what requires is biopsy in tissue and cells are taken out of a human, placed in a dish, and studied under very artificial conditions to assess oxygen consumption. And so we were very interested in could we actually identify a circulating marker that reads out systemic mitochondrial function. Now, for this study, as shown here on the left, we began with over 1,000 patients with sequence-verified mitochondrial disease and specifically asked across individuals with a variety of genetic disorders targeting a variety of different genes and pathways all within the mitochondria, are there any markers that are systemically elevated in these individuals relative to age and gender matched familial controls? And what we find is shown here in the inset box that there's over 15 molecules that are highly increased in these individuals. What we also find is that over a dozen of the molecules that are shown by the red dots here on the left all represent, once again, a biological pathway, and that these molecules are chemically very much related to one another, even though they're structurally independent. We can prioritize one such molecule and we can cross-validate these molecules as shown in the upper right here. This is a completely independent study of individuals that are controls or have localized mitochondrial disease affecting the ocular nerve without any systemic involvement, or individuals with systemic mitochondrial disease. And what you see is that individuals with systemic mitochondrial dysfunction have an elevation in this molecule. As is clinically apparent in these individuals, there's quite a variety, quite a bit of spread in the level of this marker in these individuals. And again, this is consistent with their clinical heterogeneity in that some individuals can have completely asymptomatic disease, whereas other individuals can have quite a bit of profound mitochondrial dysfunction and clinically very apparent disease. Well, we can also use this marker as a diagnostic in these individuals, and traditionally, diagnosing patients with mitochondrial disease has been extremely complicated given the over 1,000 proteins that are present in the mitochondria and the tens of thousands of potential mutations that can result in these proteins. And what we find is this singular biomarker under a formal ROC analysis has an AUC of approximately 0.8, which is far exceeds that of traditional diagnostic algorithms and or sequencing-based tools for diagnosing these patients. And this marker now is being developed here at Sapient in our high complexity CLIA laboratory as a human diagnostic for these individuals. Again, we can begin to do genome-wide association around these molecules, and in doing so, identify the key tissue, organelle, pathway, and targets responsible for this singular marker. This is a marker, just in the interest of time, I'm going to skip some of this data, but we can verify in multiple preclinical models. We can verify in cellular studies, including iPS-derived cells, as well as cells obtained from individuals with mitochondrial dysfunction, and cross-validate these in these cellular systems, as well as in other types of individuals. Now, we're very interested here at Sapient in taking rare diseases, identifying biomarkers in these rare disease conditions, and then asking, are there common human disease states or acquired disease states in which the same biomarker may also be apparent, and in doing so, be able to repurpose drugs or extend indications for drugs. Now, mitochondrial dysfunction has long been implicated in aging and age-related diseases, though it's been very much unclear whether or not these are true relationships, given, again, the inability to measure systemic mitochondrial function in individuals. And what we find now is across over 100,000 individuals in our database, these are individuals from around the world, very different ethnicities and genotypes, different geographic locations, different lifestyles and exposures. We find that this is a molecule, a singular molecule that begins to increase linearly from about age 45 to 50, all the way up to age 100. You can see an elevation in this molecule linearly across these individuals. This molecule also, independent of underlying chronologic age, holds strong association with the development of incident cardiometabolic disease, including heart failure, atrial fibrillation, and diabetes, as well as age-related tissue degeneration, particularly in the nervous system. And again, this is another marker now that begins to be leveraged not only for identifying patients at high risk for developing of age-related phenomenon, but also as a marker that's being leveraged for drug development and screening in these individuals. So I'll go ahead and stop there. I very much appreciate your time and attention over the last half an hour or so, and I would be happy to take any questions.
Thank you so much, Dr. Jain. That was a really great presentation, and I wish we had time for full discussion on the topic. We do have a couple of questions. The first question is, what are the general categories of metabolites that are detected by your mass spec system?
Yeah, so this is a really important question. Thank you for that. So there's a number of technicalities around this, and then I'll give you a very kind of high-level overview. So the way we've designed and optimized our underlying mass spectrometry systems, these are meant to capture a broad range of chemistries, everything from very polar molecules to very apolar neutral lipids, all within a singular run using mixed mode chromatography. Again, we're really limited to molecules that are below 2000 Dalton is the area that we spend most of the time our screening. But across that chemical sort of space, we're relatively agnostic to underlying chemical characteristics.
Great. Why is the focus of your system on small molecules rather than peptides or proteins?
Yeah, so it's a very good question. Now, there's some peptides and proteins that we are able to capture, but we're particularly interested in small molecules for a number of reasons. One, we're particularly interested in markers that reflect what's happening in local tissues, whether it be the gut, the liver, the brain, the heart, the fat cell, etc., but are able to passively spill from the local tissue into the systemic circulation, where we can then capture that information in a blood sample. And so small molecules have the property of being able to actively or passively transverse from the cell, from the intracellular space to the blood. And so this is a way by which we can profile molecules in blood and understand what's happening locally. Now, with proteins, as you're well aware, proteins tend to stay intracellularly, and so it can be quite challenging to actually interrogate what's happening in a local tissue or local tissue injury or disease processes through a blood sample. Now, those proteins that are spilled externally are typically done so for a particular biological reason. And we find that within the protein space, there are a number of really very important markers. They just tend to be quite nonspecific. And you can think of cytokines or CRP as sort of the classic example by which they read out many, many biological disease processes that give rise to inflammation, but don't provide great specificity across any singular disease. And so that's some of the real biological differences that are inherent to small molecules versus proteins. And we've routinely found over a number of studies, a number of therapeutic areas, that these small molecules can provide the maximum amount of predictive information, whether it be around disease processes, early diagnostics, and/or drug responsiveness.
Okay, the next question is a two-part question. How many of the molecules that are captured by your mass spec system are known versus unknown molecules? And what is the likelihood that you find a molecule of importance to you and you can't identify its structure?
Yeah, so very good question here. The great challenge, as well as the great opportunity in this space, that if we look in human blood, the vast majority of molecules floating around in your blood right now have never actually been measured or mapped or understood or chemically identified anywhere in the world. And so it's really remarkable to think about how much we understand about our genome and how little we understand about what's actually floating around in blood. And one could make the argument that what's floating around in your blood probably has more profound influence over your health or disease. Now, from any individual, as mentioned, we can capture somewhere in the order of 11,000 to 15,000 circulating molecules. Among those, we can very rapidly identify somewhere in the order of about 1,100 to 1,200. So 1 out of 15 or so can be readily identified using standard libraries that we have inherent here at Sapient. For another 1,000 or so molecules in human blood, we can computationally identify them very quickly using molecular networking, as shown in that example with NASH, which then leaves somewhere in the order of 12 to 13,000 molecules in your blood that, again, have never been mapped and are unknown and for which no standard is available. Now, among those, what we do here at Sapient is we have a variety of tools that allow us to isolate those molecules once they've been prioritized and solve their structure using both NMR and cryo-EM, and then ultimately synthesize them for verification. So we have means to go in and identify these unknown molecules. Now, the Murphy's Law of human biology or medicine or chemistry, I can't understand which Murphy it is, but almost universally, the most interesting and most important things often are the most unknown. And again, this is the great challenge, but this is also the great opportunity when you dive into the deep end of the pool. And it's really important here at Sapient that we have tools for being able to identify these molecules given their biological importance.
Okay, I think we have time for one more question. What is novel about your mass spec system other than going fast?
Yeah, so good question. So there's, mass spectrometry is something that's been around for 40 years. So by no means did we invent the mass spectrometer. The way a mass spectrometer operates is through coupling to a separation technology, typically called chromatography. And really what's happened over the last decade or so is that there've been a number of advances in high resolution and IMS-based mass spectrometry, but that upfront chromatographic separation really hasn't fundamentally changed in the last 30 to 40 years. And so here at Sapient, what we've done is we've developed new chromatographic separation systems, again, that allow for capture of very different molecules all at the same time, and allowed these chromatographic systems to operate very quickly so we can actually leverage all the advances that occurred in mass spectrometry. Now we've been able to couple these chromatographic systems with IMS high resolution QTOF mass spectrometry, as well as develop the back-end software that allows for machine learning based rapid image sort of processing, so we can handle tens of thousands of mass spectral files, both in isolating and extracting data, as well as aligning these files across many different individuals in a given study and across different studies. And so this is what allows us to go very quickly to measure many molecules, as well as to have done this across hundreds of thousands of individuals in this underlying database.
Great. Well, thank you so much. On behalf of AAPS, I do want to thank you, Dr. Jain, and the whole team at Sapient for a great presentation. For those of you who are joining us, this has been recorded and will be available on the AAPS On Demand webinar site. Thank you again, and we look forward to seeing you at a future presentation. Have a great day.
Thank you so much. Have a wonderful holidays.
You too.
Bye-bye.